Ladies and gentlemen, good morning. Thank you for your kind introduction. I'd like to thank Professor Veronesi and Professor Chiara Tonelli for their kind invitation. Actually, the list would be much longer. There are indeed many friends whom I'd like to thank, but for the time being, I believe that we should also and always start these days of work stating that these are very important meetings, the one that we are holding today for a country such as ours, to try and stop the decline of our country. So I think that we should be most grateful to Umberto and Chiara for this enormous and wonderful effort. As for the title of my presentation, well, you've already heard of it. Professor Chiara Tonelli is really sweet, or looks sweet, but she's very bossy. So I understand that my time constraints are very tight. I've already done with a minute I had. I've already lost a minute. So I'll move on. There are only images that I'm going to share with you so that you can have the opportunity to catch the point with this nonverbal communication so that I can go straight to the point. You know that there are athletes that run very fast to get to the goal. Now the second part, so I'll be cover cloning and I'm going to again go through a general overlook of cloning, and then I'll be concentrating on stem cells. And I can benefit from the fact that immediately after me, Professor Costa is going to take the floor, and he's one of the top experts globally on the subject. So whatever I neglect is going to be covered by Professor Costa's presentation shortly afterwards. You heard a previous speaker, and especially Professor Testa, state that what is important is that language could set up traps when information are transferred over. There are problems connected to genetic epistemology that are to be highlighted in italics, in bold characters, so as to be crystal clear. Historical background is also important. Biology is a science of the description of what is living. It has now become the history of whatever is living. Whenever we talk about cloning, we're not talking about anything new, because on a daily basis, cloning is being experimented with plants. We all know that clones are amongst us, homozygotic twins, for example. And if you've ever seen a very nice animal such as this, whenever the mother gives birth to her litter, there are always 4 clones. And some animals actually even do that on a larger scale and make as many as 8 clones. So can we actually reproduce artificially? Yes, we can. Vets are always one step ahead of biologists and general practitioners or physicians or medical doctors. So I see some friends that are, showing off because they have this time advantage. Embryo splitting is a technique that has always been used in veterinary science. I should also like to remind you that when we talk about historical background, we always have to refer to Spemann, that was awarded a Nobel Prize in 1935, and in 1938 he developed this idea of the fantastical experiment. Now, I cannot dwell on the detail of Spemann's experiment. We already talked about that with Luca Ronconi in Spoleto. when we were talking about the life of the assistant to Dr. Steyman that Mangold, because it was Mangold's work that actually contributed to Spearman being attributed to the Nobel Award, but unfortunately she had a major problem during her lab life. And go to the Nobel website, because you will find all the talk of all the Nobel laureates. So this is really important website. Spearman was charged with accusation of being a Nazi scientist, which is completely misleading. Spearman goes from Berlin to Freiburg so as to support his German friend to leave the country. He's a scientist. He has a big idea. He was thinking, if I succeed in the shifting a nucleus through it cytodifferentiation in the cytoplasm of the cell of a man, then I may perhaps reprogram the embryo development. At that time, Dr. Stamens didn't have the necessary tools. As a matter of fact, he could only rely on hair. And there's a wonderful description of the hair of newborn children, especially so of one of the child of Mangold, the assistant to Spearman, that the hair was very soft. And he used these hair to perform ligature on eggs. And then there's also an important description of the life of Mrs. Mangold, who's the first assistant to Spearman, and then Mangold was the fiancée of Hamburger, but then she leaves him and got married to somebody else. And then that man met with Rita Levi-Montalcino. But I strongly recommend you should visit the website of the Nobel laureates. Clones were developed thanks to the assistance of Mangold, but he couldn't succeed in the cloning. It was only in the 70s that cloning was successful. This is the first intercytoplasmic sperm injection that was developed by Ryuzu in Honolulu. And this is an image that is to be found in the history of the university in Pavia, because this was given as a gift during a ceremony when laurea honoris causa degree was granted. An important development took place technologically wise, because here there's a needle to penetrate that big cell, the egg, and there was also small test tubes with the pressure of rubber tube that was held in the lips, so that the scientist could either blow in or suck, so as to inject, to transfer basically the component into the cell. And that allowed for a transfer of nuclei as Spearman had thought. I'm not talking about the hybrids in the transfer of cytoplasm. Yet you can easily see the two theoretical lines. So on the one hand, you can transfer nuclei in oocytes, so as to obtain the reconstitution of a single cell embryo, or the zygote, and then you can have the development of a clone. And if it is only stay in the test tube, it'll produce embryonic stem cell. Now, this is only a very rapid overview, and to present again the different steps of cloning. So amphibious elements were first used. John Gordon is indeed the first who obtained a good percentage of clones being born And this is the frog that again was the subject matter of this experiment. You should really look up this website. You are, and I'm addressing the students amongst you. You're really, really lucky. You have wonderful teachers, and this is so important for your lives. And perhaps you can even be awarded a Nobel Award, but perhaps your teachers might not appreciate your work. In Cambridge, for example, John, in his desk, keeps here's a school report at the end of the high school when he was asked what he wanted to do. He says he wants to do biology, but he ranked 52 out of 57 students, and so as you can possibly read in this document, he was told that it would be quite ridiculous and it would be an utter waste of time for you and your teachers if you were to take biology. So, and he was awarded a Nobel Prize. So just remember that. It was only 1981 that the first mouse was cloned. Carl Ilmenzi and Pot Hopper, an American scholar, succeeded in that. And again, caution is always required. This is the first case of fraud. Why is that? Well, because actually nobody succeeds in repeating the same experiment. There's an investigating committee with the Salter leading it, and he is working in Heidelberg. And he completes his investigation, stating that cloning of a mammal is utterly impossible. So Carlo Ilmenzi loses all. But I believe that it was not a fraud. Together with others, I wrote a letter so as to give him a new status, but this unfortunately did not happen. I believe that much depends on the skillfulness. Perhaps the techniques were so vulnerable and fragile that only a happy few could succeed. So more time had to go by. It was only in 1984 that Willesden obtained clones. And then in 94, and then Neil First, and 96, Ian Wilmot and Keith Campbell. Ian is a strong biologist, but he was actually a vet. And the one that actually carried out the work is a friend that died very young, Keith Campbell. And Keith Campbell actually is to be acknowledged for this because he was the one that actually manually succeeded in carrying out this experiment. And then this is the history that is known to all and everybody. The next year, in February 1997, Dolly was born. At that time, the technique was perfectly well developed. Now we have quite well sophisticated tools. For example, the movement of these needles are performed with piezoelectric systems, and the development of tools plays a very important role. We've learned about that also yesterday. So we cooperated with the Honolulu School, and that cooperation had started quite a few years ahead of time because we were trying to keep vital some sperm that were not preserved in refrigerating units. So we performed the Spearman's experiment. Dolly, is not Spearman's experiment, is an important advancement, but is something else because the two cells, through a virus or through an electric shock, the two cells were actually merged together, so it was not Spearman's experiment. We decided to carry out Spearman's experiment. We tried with different cells, then we performed the experiment. This is the Honolulu collaborator, and this is Cumulina and her foster mother. You see the black mouse is Cumulina, and the white mouse is her foster mother. As you may easily understand, the press covered the story extensively, but the first thing to be stated is that cloning is a highly inefficient event. You see that Rizuya Nagimachi, the Honolulu doctor, pointed out to the fact that clones are sometimes fertile. Cumulina, for example, turned out to be fertile, and there's been a sequence. And a whole zoo was set up. You probably remember the bull Galileo Cesar Galli was even arrested because apparently what he did was considered as impossible. There was a law adopted by Italian Minister Rosy Binti that banned that. And then Snoopy is the result of another experiment. This is Su Wang, another major scientist, that carried a biologist that was charged with different accusations. Stamps were devoted to him, but at some stage it appeared as if he was the author of a fraud. Now, for a whole set of reasons, which I won't dwell upon, he exaggerated a bit the yield that he had with his stem cells as obtained with the oocytes of her students, and this is an infringement of the existing legislation. He probably wasn't aware. So because he had his scholars, his students involved, and that was against the law. So the result of his experiment was considered him as a fraud. But the press, certainly not Beccaria and not the Stampa, the Italian newspaper, were not to be considered as guilty for what happened to this doctor. As a matter of fact, you cannot involve your students. Just think that, for example, Dolly is 1 out of 486. With Cumulina, we had 1 out of 84 surviving. But this, again, is a mark of the major inefficiency. For example, there are dilated organ syndrome. The clone usually is born pretty fat, perhaps even obese, and there are important problems to be tackled. Now, I'm going to make a long story short because I cannot explain the theory of complexity, but the clone usually is more prone to being dumb, sick, and obese than its ancestor. So a lot was written on cloning. Well, you have to understand that this is a crime. You can be sent to prison for 10 years, to serve a sentence for 10 years if you perform cloning. And then there is another important piece of evidence. We are not that good, so there's no problem, no alarm. You know, the children coming from Brazil with blonde hair and blue eyes is something that we cannot do. So, and we can't do, as a matter of fact. When Professor Veronesi in the year 2001 asked me to sit in the Trebecki committee, I was strongly supported to support this cloning was Cardinale Tornini, the cardinal. Was explained a lot. I cannot explain you who Rael was. Rael is the gentleman sitting next to Brigitte. Brigitte was a very good biochemist from Paris, but she left the university when she was found out as being the fiancée of Rael. Le Monde had wonderful comics on that. There's a lot of fear of cloning, but such fears are to be left behind. So there's an important technique, and we have to make a distinction between the technique and the result it brings about. So if you can clone a sheep that I obtain as Dolly, and I can obtain milk out of that clone, and I spent €1 million to have this transgenic sheep. If I cannot obtain any milk, there's no point in having another €1 million to have the same transgenic sheep. The technique is certainly important because cloning is the evidence that the cytoplast, that is the oocyte without the nucleus, can reprogram, can be reprogrammed genetically. And as you see in this, we can actually verify where we find stem cells, and now I think that pluripotent is only the zygote, and the zygote is actually produce all the cells that make up our body. That limitation in power in the differentiation of cells, whereby we go from pluripotent, and pluripotent are all the cells of the embryo down to the time when it is implanted in the uterus mucosa. So there are embryonal cells. After that, from fetus onwards, after the implanted embryo, there are somatic cells. Those are that the press defines as adult cells. And then there are single power cells, as the one that make up our hair. I believe that this sketch is very clear. We can reprogram cells in that we know we are familiar with the stemless genes, and we can push an undifferentiated cell and reprogram it. But these are just different changes in the Woblington landscape. Epigenetics here plays a role. There are different landscapes. There are peaks and valleys at times of stability of the cell, but the use of metaphor is pivotal while we are ignorant. And this is something we have to stress quite clearly. Then we can also consider as something that I don't like the wording of, and that is therapeutic cloning. Now, I believe that I have 10 more minutes, and with these 10 more minutes, I'm going to show you that I can use this computer. Maybe I'm not that good, though. Done. You know, as when you go to the doctor, basically you don't feel any more symptoms. We're going to hear Professor Kossu on this subject. What I'm going to do is tell you something else. So this technique can certainly be used to produce embryonal stem cells, but we have to make a distinction from the technique to the product of that technique. This is fundamental. With these cells, then different treatment can be administered. We made important progress on that. Just think, for example, about blood tumors. I think about the major burns. I think about, for example, De Luca with the bank of the eye. From one cell, he can actually reproduce corneas. We've made also a lot of improvement to treat myocardial infarction. 20% of the pump function is recreated by the left atrium, and what is needed is just heart ultrasound and a needle. So there are evidence showing that these cells can be perfectly well integrated, as is shown in that chart. We've done an important also improvement to treat diabetes, type 1 diabetes. Now, we believe that with insulin you can treat the whole disease, but as a matter of fact, you cannot heal the diabetic patient. You can just treat one of the symptoms. We've made important progress also for Parkinson, not for Alzheimer and other degenerative diseases where there is an architecture of the tissue that has to be replaced. But if I have to change just one brick, for example, the dopaminergic neurons of the substantia nigra, then this technique is good. Controversies emerged, for example, the leftist papers such as The Guardian wrote incredible things, completely wrong things indeed, and this was due to the fact that the operation that they did in the US was wrong. Now this gentleman has invented this special needle that enables you to go through the ear, coming to the substantia nigra with no problem whatsoever. Then, you know, obviously a problem still is finding these cells. So you can shift from one embryo cell, and out of that, 1,000,000 cells, i.e., as many as the dopaminergic neurons. Why can't we treat Parkinson's disease? Well, because a judge in Europe and European judgments, cannot be changed. No, when a European judge states something, that remains unchanged. So Greenpeace is certainly to be protected for what they did for whales, but this judge, and he has 5 degrees of veterinary philosophy, biology, and so on and so forth, and he stated that one cell is a human embryo. Well, if a student tells me something like that, I sent him home. But unfortunately, this judge pretended he had 5 degrees. Unfortunately, he knew nothing. But unfortunately, the judgment he passed was not to be repealed. Now, you all know that all human beings are mortal. Socrates is a human being, then Socrates is mortal. But there's an incorrect faulty syllogism. All birds sing, Eminem sings, rap songs, so Eminem is a bird. And this is a bit what happened, This is a report that was published in the US, a very interesting report. You can certainly read it at ease. And what it says is that all that embryo are innocent, so killing an innocent is a crime, is murder. But what is important is to understand who's who. We cannot just use such syllogisms. We've tried on a little scale with Schöller and others. And, we stated that if there is constitution of such embryos goes in a culture terrain or in another, in that case was ZB as opposed to alpha mem, then the results are different. So we cannot say that embryo stem cells are not embryos. I'd like to remind you 2 final things. One, the technique is very important, and in the UK now, they are showing they're being democratic. We're not talking about Tony Blair or Cameron, but there's true democracy because they're asking people where to go. So, for example, the technique in this case is proved to be effective to transfer nuclei. And that technique can be used to prevent mitochondrial disorders. So money is spent on informing the public. Some sort of referendum is held, and the result is that that is feasible. So if 2 children exchange the oocytes, the 3-parent can be established. So as you see here, you can do away with the mitochondria that cause devastating disorders in children of 8 and 2-year-old. The 3-parent is just because there is a contribution from another woman producing or offering her mitochondria, but these are just, elements producing energy. Augusto Pessina wrote a science that is ruthless. So this is not the case, and I criticize him. I think I should have royalties because I talked about this Habermas, especially this book by Habermas. I spoke so ill about this that I should be given money because basically I advertised it. But this is something that completely impossible. Shut up your molecular biology lab because these people, mix things together, and this is absolutely unacceptable. While we were here over the past few days, we've been asked what we can do in terms of sperms of oocytes that can be termed as artificial, because I have stem cells on the one hand. I can use chemical factors in the culture so as to become sperms and oocytes. then I won't talk about the medical applications, but we have a new source basically of sex cells that are extremely potent in providing sperms and oocytes. As for the sperm, well, the sperms were used in the naked the mouse was used in these tests. Then oocytes can be generated. I am for open source, so if you want to use these images, please do that freely. For example, with mouse, we can obtain fertile animals, and this is done by many groups. Obviously, we've got to talk about that. Now, we have the possibility of doing that in men in the States. A scientist did that for men without sperm. There are certainly aspects that are to be debated. Together with Monti, we worked on these cells. These are wonderful cells that were developed by Manuela herself. And taking from the code without any ethical problems, but taking small cells that are called very small embryonic-like, VSEL, we tried to becoming oocytes. Here you see some markers of stem cells. These are the embryo bodies that are created in the test tube, then it is disgregated, and then you can develop male eggs and female sperm. I understand that my time is up, but just Chiara, please, one more minute and then I'm really done. Obviously, this leads you new parenthood. This is Isabella Rossellini with Madonna in a very famous snapshot, and you're also very familiar with this one. And this is another system that well, this happens in bed if everything is all right. This is what happens in bed. Now, the take-home message for the boys. Ours is a species with internal fecundation, and the female can discriminate against. As for flowers, you have to send them after. First, you have to invest on dinners and taking your female out. But this is the beginning of a new individual. This is not the beginning of life. Life started over 3 million years ago. This is the autogenetic beginning of a new individual. If you're depressed, you have to think that you've come before others. But there's a fundamental element for the philosophers, and we need, philosophers. We strongly need philosophers. We're very close to Massimo Cacciari, a philosopher in Venice, and I've often said to Massimo that they shouldn't talk about the apple of Newton. Philosophers should think about biology. The male is redundant. You don't need male for reproduction. I see that the boys are worried and the girls are happy, and that's quite right. And Hannah Rosen stated that this is the end of man. I saw something very strange, carried on a bike like that. What is important is to decide where to go. You have to, and perhaps we should talk, as they are doing in the UK, whether to pay or not, because leaving the Columbia University after a seminar, I looked at the paper and I saw many ads, and one among them was this ad where girls basically are invited to sell their eggs, and she can earn quite a lot of money doing that. But a geneticist stated that he is willing to pay 25,000 if the girl, as a master, she's got blue-eyed and she's of a certain age. This is a shop in Honolulu. But this is not a true or Genesis boutique. They just sell clothes. You know, we have to think about the health of women. As for ethics, well, we don't have to be biased. We don't have to talk about religion and ideology. Certainly, those biases are quite legitimate, but we have to be able to shoulder our responsibility and make choice. As for frozen embryos, well, Pope Ratzinger, talking to Habermas, stated, you know, the damage is caused and it cannot be corrected. Well, I'm an atheist. I think that we have to respect religious people, and Cardinal Tonini stated as much. Everybody obviously has to be involved. What is important is to pass a decision. Each of us should be given the tools to make the decision. Gabriele stated that the scientific citizenship, for example, you should forget about Jefferson and the American Revolution. You should forget about the French Revolution. Now, you all can be considered a citizen if you know about these things and if you can express your opinion. So we cannot but thank the Fondazione Veronese for disseminating so much knowledge and for this important advance in culture that they promote. Thank you.